You hear me do it a quarter before, it's gonna basically be the same lecture, okay, with minor differences, but it'll basically be the same thing. And I'm sure that I emphasize the same things, I'm sure I said 10 times in that lecture, there will be the renin-angiotensin system on your phone, I'm sure I said that. Okay, so what I wanna do today is, there's 2 or 3 slides from the urinary lecture that we didn't finish. It's in that lecture that I posted, but I figured out what the heck, let's just do it, okay? So we'll do that. Basically, we're just looking at the urination reflex is what this one is. So we'll go over that. And then we're gonna start fluids, electrolytes, acid-base buffer, okay, that PowerPoint. And it'll take us 2 lectures. I'm hoping a lecture and a half, not 2 full lectures, but we'll see what happens. When you go to start studying for that, there is a worksheet posted, I think under study guides. You should do it. It's really helpful to just kind of work through those questions. It'll help it to get in your head for sure, okay? So, I do those. Obviously you don't have to, but I would do it. Don't forget that you've got things due. I think you have something due today, yeah? Is that true? I think so. And then the other thing is due on the 3rd, thank you, on Friday. If you couldn't get what was due today, if you couldn't get it in, let me know and I can change that on Canvas so that you can still get it in, okay? Those 2 assignments should be easy 23, 24, 25 points per assignment, okay? I rarely have somebody get below. Sometimes I'll have people get 20, 21, 22. But if you were to get lower than a 20, I would talk to you about it and see about maybe having you redo it if you wanted to do that. But let's just try to avoid that, okay? Because you don't really have time to be redoing things. You got a lot to do, okay? We've got 2 weeks before the end, so we have a lot to cover. All right, I'm gonna stop rambling. Questions, comments, issues? Did you have a good Thanksgiving? Yeah, you all look so excited to be back today. So excited. It's not raining, you should be happy. All right, I'm gonna take a drink of water and then we will get going. Tried to go down to Olympia for Thanksgiving, so we left like really early in the morning. Stopped at 5, like 7 o'clock to get gas and a cup of coffee at Starbucks. We came out of Starbucks, we were in there maybe 7 minutes, maybe. Came out of Starbucks and somebody had bashed in our driver's window, just destroyed it, shattered it. So we had to go back home. It was very annoying. It was not very nice. Don't go to fight. Didn't steal anything, that was good. We had a cell phone and a bag full of money and an iPad was sitting right in the front seat. Didn't think anything. Weird. Okay. All right, on that positive note, let's get going. All right, create. Have you guys heard anything different about winter? Scheduling classes, anything different? No? Just curious. There's classes you can take in person in the winter? Are most of them in person or online? Mix? Alright. Okay. And here we go. Stretch. Okay, so just to finish up the urinary system. So we had left off getting the filtrate through the tubule, and we had ended with the DCT and the collecting duct. And once that filtrate leaves the collecting duct, then it is urine at that point. It's not filtrate once it leaves the collecting duct, and we can't do anything to it anymore. We can't add to it, which would be secretion, and we can't take out of it and put into the blood, which would be reabsorption. That filtrate is going to that urine is going to first collect in the minor calyx, then it will drain into the major calyx, and you can even see it on here. So the minor calyx would be like here at the base of those renal pyramids, and then it would drain into like a major calyx would be kind of like here and there, and then from the major calyx it would drain into the renal pelvis, which would be, it shows right here, it would be right there, over here would be about right there, and then it's going to leave each of the kidneys through the ureters, collect in the bladder, and the whole purpose of the bladder is just to store that urine until we're ready to eliminate. And then we will eliminate by the urine leaving the bladder and exiting the body through the urethra. Now, in order to get it out of the bladder, we have to have a urination reflex get triggered. And that's what we want to look at here. So a urination reflex, also called a voiding reflex, or it's also called a micturition reflex. Okay, micturition reflex. Involves both voluntary and involuntary aspects. And this urination reflex or voiding reflex is very, very similar to a defecation reflex. It's pretty much the same basic idea. So what we have, when you look at the urethra, you have an internal urethral sphincter. And sphincter is just another word in this case for valve. Okay? And then you also have an external urethral sphincter. Now, these are not at the very end of the urethra, okay? They're actually closer to the bladder. And you can't see this on your models, okay? You can't see it on the model. But these are terms that are on your lab supplement, so for your lab practical. And though you can't see them on the model, what I want you to know for lab, and then we'll talk about them for lecture here, is what kind of muscle they consist of, okay? So your internal urethral sphincter consists of smooth muscle, and your external urethral sphincter consists of skeletal muscle. And if you were looking at the defecation reflex, you would just, instead of calling these urethral sphincters, you would say internal anal sphincter and external anal sphincter. And it'd be the same way, smooth and skeletal muscle. Now, at a resting state, both of these muscles are contracted. So right now, they're contracted. And that helps to keep the urine in your bladder instead of kind of dribbling out of your bladder and going through the urethra. So these guys are closed. And that's why I say it's up close to the bladder, because as they close, it helps to keep the urine in the bladder. If they were, particularly think about a male. The male's urethra runs through the penis. If you put those all the way at the end, then you would have urine sitting all through the urethra at a resting state. You wouldn't want to do that. Particularly for males, because the urethra is a common passageway for semen and for urine. We'll talk more about that when we do reproduction. So these are closer to the bladder, towards the top of the urethra is where they are, okay? So at rest, they're both contracted. Now, for voiding to occur, you've got to have basically three things happen. For voiding to occur, you need each of these to relax, okay? So you need, for voiding, you need this to relax. You need this one to relax. And you need to have the pressure. I know my marker's working. You need to have the pressure in your bladder go up, okay? Because urine, like blood or air, is gonna move from high pressure to low pressure. Okay? So if we want that urine to exit, we need the pressure in the bladder to be high so that it will allow that urine to leave, moving from high to low pressure. All right, now, when we look at this reflex, remember reflexes are automatic responses to a stimulus. That's what a reflex is. Some kind of motor or glandular response to a stimulus that's automatic, meaning you don't think about it, okay? And the part of this that you don't have to think about is the part that deals with smooth muscle, and the way that we increase the pressure in the bladder is by making smooth muscle in the wall of the bladder contract. So let's look at the pathway that regulates that part first, the part you don't have to think about, okay? And then we'll talk about the voluntary part of this process, the part that you have some control over, okay? We'll do that afterwards. Okay, so we've got to have a stimulus. That's going to start the whole thing. And that stimulus is urine entering the bladder. As you get more and more urine entering the bladder stretches more and more and more and more and more. So the more urine that's in there, the more the bladder stretches. And in the wall of the bladder, you have stretch receptors that detect that stretch. Once you get a certain amount of urine in the bladder, then those stretch receptors become active enough that kind of triggers this whole pathway to start being engaged. And that usually is around 200 mils. And it depends on the person, but it's about 200 mils or so is when you get enough stretching and enough activation of the stretch receptors that this thing goes. Now, what's going to happen is the stretch receptor is going to be activated. That then is going to cause a signal to be sent from the stretch receptors in the bladder to the spinal cord. This is automatic, so it does not have to go to your brain in order for the involuntary part to work, okay? Which would be the things that don't say voluntary on there, okay? So this information is going to go to your spinal cord, right? It goes to the lower part of the spinal cord, and in the spinal cord, this information is going to be received and processed, and coming out of the spinal cord, going to the smooth muscle in the wall of the bladder, and going to the smooth muscle of the internal urethral sphincter. You're going to see an increase in parasympathetic activity and a decrease in sympathetic activity. Okay? So stretch receptors become active, information gets sent to the bottom part of the spinal cord. Coming out of the bottom part of the spinal cord, we will see an increase in parasympathetic, a decrease in sympathetic, going to the smooth muscle of the internal urethral sphincter and the smooth muscle in the wall of the bladder. Okay? Now, having more parasympathetic and less sympathetic at the internal urethral sphincter. Let's put that up there. So, I'm going to see more para, less sympathetic here at that smooth muscle. What this is going to do, actually put it here, at this smooth muscle, what that will do is it will cause relaxation of the smooth muscle and the internal urethral sphincter will open. same time that's happening, having that increase in para, decrease in sympathetic, going to, okay, again, that's coming from the spinal cord, going to this smooth muscle in the wall of the bladder, that's gonna cause this smooth muscle in the wall of the bladder to contract. So you got two sets of muscle receiving the same signal. One set of smooth muscle relaxes, one set of smooth muscle contracts. And that can happen because even though we've got the same neurotransmitters being released, we have different receptors at those smooth muscle. Over here, we're activating inhibitory receptors. Here, we're activating excitatory receptors. Well, that's all. So here, this smooth muscle starts contracting. That causes the pressure in the bladder to rise. All right, so the only thing we have left to do is we have to make this smooth muscle, this skeletal muscle, we have to make it relax, okay? So we've achieved two of the three things so far. Now, to get this skeletal muscle, to get it to relax, now we've got to involve the brain, okay? So at the same time, that sensory information is going from the receptor, the stretch receptors, to the spinal cord to cause this reflex to kick in. Relaxation here, contraction here. As that's happening, that sensory information is also going up the spinal cord. So it doesn't just stop in the spinal cord and cause this to happen. We also have another pathway that's heading up the spinal cord. And that sensory information that's heading up the spinal cord heads up through the spinal cord, up through the brain, synapses along the way, but eventually makes it to the top part of your brain. The top part of your brain is called the cortex, okay? So your sensory stimulus gets up to your cerebral cortex, that's the top part. And that's the part of your brain where if information gets up there, that's where you have aha awareness, okay? Only do you have awareness if information comes from or goes to the top part of your brain. So that information that went to the spinal cord and right back out to the smooth muscle, you don't have awareness of that pathway. But because the sensory information also went up the spinal cord and synapsed in the cortex, that's when you say, oh, I need to go to the bathroom, right? And then you decide if you're gonna act on that urge to void or not. If you decide to act on that urge to void, then what you are essentially doing is you are causing those descending voluntary or somatic motor pathways. Descending somatic just refers to skeletal muscle. Somatic, descending somatic or descending voluntary motor pathways to the skeletal muscle of your external urethral sphincter. You're causing those to decrease. Normally they're real active, and that's why normally this muscle's contracted and that sphincter's closed. So as you're sitting there, that pathway is nice and active and that sphincter is closed. But if you decide you're ready to void, you are causing decrease in those action potentials. You're causing less action potential to travel along this pathway. And this pathway, because we've got somatic motor going to skeletal muscle, this pathway is releasing acetylcholine, right? That's your neuromuscular junction that you learned about last quarter. So if you have less action potentials traveling along this pathway, you release less acetylcholine. And if you're releasing less acetylcholine at those neuromuscular junctions, then that muscle is going to start to relax. So that causes relaxation. And as you have relaxation of that skeletal muscle, that will cause your external urethral sphincter to open. And now we've achieved all of these things. We've got the smooth muscle relaxing here. We've got the smooth muscle contracting here, increasing pressure. And now we've got our skeletal muscle relaxing as well. And then having high pressure in the bladder, low pressure outside, and those two sphincters opening, now urine can exit. But if you decide, I don't really want to act on that right now, I don't want to act on that sensory stimulus. If you say, now's not a good time, then you will not do this. You will not cause these pathways to slow down. And if you're not causing these pathways to slow down, then they are still nice and active. So they're still active, which means you've got plenty of action potential traveling along them, which means you're releasing lots of acetylcholine is being released at those neuromuscular junctions. And that keeps that skeletal muscle contracted like it normally is. And that keeps your external urethral sphincter closed. Okay, so even though this muscle is relaxed and this sphincter is open and the pressure in your bladder is rising, you can say, nah, I don't want to do this right now. And keep that closed. And you can keep ignoring this urge to urinate for a certain amount of time. But once you get around 500 mils or so in your bladder, the problem's going to be that the pressure in your bladder is going to start to get really high. Because not only are you triggering this reflex with all that urine in the bladder, that's triggering this reflex, the smooth muscles contracting, but now you just have a lot of urine in there, which is also increasing the pressure. And at a certain point, if that pressure gets too high, it can force this to open. It can force this to open, even though you're trying to keep it closed, right? And certain things can cause it to be harder to keep that closed. If you start laughing, right? If you start laughing and you have to go to the bathroom, a lot of people will just go to the bathroom right there, whether they want to or not, right? And that's because when you start laughing, you're engaging your abdominal muscles, and that's squeezing on your bladder, and that's making the pressure go even higher. Sometimes, if you have to go to the bathroom really bad and you're getting close to the bathroom, like you're putting the key in the door, right? That other cerebral activity can start to interfere with this, so you can't keep it closed quite as well. When you start seeing the bathroom, if you have to go really bad, or if you start hearing water, all those things can start to interfere with you keeping those descending somatic motor pathways nice and active. As you get older, as people get older, you lose neurons as you age. And because at a resting state, neurons being active is what keeps these guys closed. As you age and you start losing neurons, you have less of an ability to keep these closed. And that's where incontinence can come into play with aging. All right, questions on the urinary system? Okay, I'm gonna get out of this PowerPoint, pull up the next one, take a sip of water, and we'll get started.
The green one.
I'll leave that up there for a minute. You want me to clarify something up there? I know I scratched them. Can you read it? Can we clarify anything?
Still active.
Better? All right, take a picture of it and we'll take the water. And then I'll get started on this. All right. Okay, fluids, electrolytes, acid bases. A little bit of this, a little bit of that. Nice thing about this PowerPoint is some of the things that are in here we've already talked about. We've already talked about either with hormones and or with the urinary system. So for some of these things we'll kind of breeze over. For some of these things we'll review and then add more to. And then we're also going to learn some brand new things in this PowerPoint as well. So in this PowerPoint, where we'll spend most of our time is talking about buffers. That's where we'll spend a lot of time, and we'll talk about acid-base disorders. Okay, so we'll get to that after we do some other things first. So we're going to first talk a little bit about fluid. I'm not going to talk a lot about it, but there's a couple things that I definitely want to review and make sure that you're comfortable with. We're not going to spend a lot of time on electrolytes. Again, there's a couple things that I want to review and make sure you're comfortable with. We're going to spend the bulk of our time with acid bases and buffers. Okay, so I will make a point of letting you know from some of these slides what things you do need to know, what things you don't need to know. Okay, so for the first few slides that are looking at water, what I definitely want you guys to kind of keep in mind is when you look at your body weight, most of it is due to water. If you think about a cell, right, just think about a cell. Inside that cell is the cytoplasm. And that cytoplasm consists of the cytosol, which is the liquid part, and then structures or organelles. And that cytosol really is mostly water, and then just whatever's dissolved in it. That's your cytosol. So inside of your cell is a lot of water. And then if you look outside of your cells, if you look at your extracellular environment, that's mostly water too, with things dissolved in it. So you've got cells in these aqueous environments. Just the fundamentals. They're little chemical factories. They're just constantly doing chemical reactions. All the time they're doing chemical reactions. Even when a cell's just at rest, it's making ATP, it's using ATP. It's making proteins, it's making carbs, it's making lipids, it's breaking things down. It's constantly doing chemical reactions. And remember, to do chemical reactions in our body, you need enzymes. Even if the chemical reaction is a spontaneous reaction, meaning that it will occur on its own, even if that were true, it's too slow. Your chemical reactions, even if they're spontaneous, are just too slow to sustain life. So that means basically all of your chemical reactions, whether they're spontaneously occurring or not, require enzymes. They all require enzymes. Enzymes catalyze reactions. They speed up the reactions so that they happen fast enough to facilitate life. They also do some other things, but we won't get back into enzymes all over again. But if you think about an enzyme, they're proteins. Enzymes are proteins. And proteins, as are enzymes, are water soluble. So they are dissolved in this aqueous environment. So most of your chemical reactions are dealing with molecules, substrates, enzymes, products that are water soluble. Not all of them. Obviously you make lipids, you use cholesterol. Those aren't water soluble, but most of the things that you're doing in terms of chemical reactions are dissolving in that water. So water is pretty important for that reason alone. All these reactions are taking place in this environment of water. Now, also, water can freely cross any membrane it comes in contact with. It can diffuse faster if there are water channels present, if there are aquaporins present. But even if there are no water channels, if there are no aquaporins, water can still freely diffuse in or out of a cell. And what we want to avoid are cells gaining or losing too much water. So that's another reason we're really concerned about water. We don't want too much fluid shifting in or out of our cells. It can be dangerous. So, when we look at homeostasis of fluids, right, what we want is, we want to make sure that, again, our cells aren't gaining or losing too much water. And remember, water will move via osmosis towards a high concentration of solutes. Okay, let me write that up here. Osmosis says, and here we're talking about just plain old osmosis, not dealing with plasma proteins specifically or the blood specifically, just in general, when you look at diffusion of water, osmosis. Osmosis says that water will move towards a high concentration of solutes. So water's going to move from low solutes to high solutes. Okay? Or you could say water's going to move from areas where you have low solutes by water towards areas where you have high solutes, low water. That's where water's going to move. Okay? It's going to move towards areas where you have low water, high solutes. So, that's also why we're concerned about electrolytes. Where do we have a high concentration of this ion versus that ion? Do we have a sum total of equal solutes in and outside of the cell? And I'll get into that more in just a second. So here we're looking at extracellular versus intracellular, and we want our extracellular environment and our intracellular environment to be in homeostasis with one another in regards to total solutes. Okay? Total solutes in and outside of cells need to be equal so that water is not rushing in or out of a cell, moving towards an area of high solutes. Now, if we look at individual ions, they can be high concentration one place or another. But when we look at the sum total of solutes, they need to be equal in versus out, so we're not gaining or losing water. Okay? I'll say that a couple more times in just a minute. So, extracellular, everything outside of cells. Okay? There are different subdivisions of your extracellular compartment. So your interstitial fluid is part of that extracellular environment. Your blood plasma is the liquid part of your blood. It's outside of your blood cells. That's extracellular. Things like your synovial fluid is extracellular. Your CSF, cerebrospinal fluid, that's extracellular. Any fluid that's outside of cells is part of the extracellular environment. And then inside of your cells, you have your cytosol, okay, your cytosol. And you have trillions of cells in your body, and when you start adding up the cytosol and all of those trillions of cells, it ends up being quite a lot of fluid. One individual cell, there's not a lot of water in the cytosol, but sum total of all your cells' cytosol, that's a lot of water. All right, we'll come back to that. Ions, electrolytes. You don't want you want to make sure that certain ions stay in their homeostatic range. So for example, we've talked about blood calcium levels, how critical it is that blood calcium levels don't get too high or too low. They need to stay in their homeostatic range. We've looked at things like potassium. We don't want potassium levels getting too high or too low. We need them to stay in their homeostatic range. Sodium levels, like blood sodium levels, for example. All these various ions, particularly calcium, potassium, and sodium, if they get too high or too low, those three in particular can start to influence things like muscle contractions, action potentials. You can start to get heart arrhythmias because your heart muscle isn't contracting properly. Those all three are ions that we talk about when we talk about a cardiac action potential. Sodium, calcium, potassium. pH. pH is pretty important too. pH is a measure of hydrogen ions. If we have too many hydrogen ions floating around, then our pH starts to drop, and that's not so good. If we have too few hydrogen ions floating around, then our pH becomes too high. That's not so good either. Typically, we deal with low pH versus high pH because our cells make more acids than they do bases. And so typically, we're dealing with, oh, the blood pH is a bit too low, let's fix it. Okay, again, we'll talk more about that. The reason pH can be so problematic is if the pH is too low, let's say, that can start to interfere with hydrogen bonding. If we start interfering with hydrogen bonds, now we start to disrupt the 3D structure of proteins. And if we start to make our proteins denature, and we can make proteins denature by cranking up the heat, but also changing the pH, in particular lowering that pH. If the pH starts to lower, the hydrogen bonds get disrupted and the protein can denature. And if we start denaturing proteins, cellular homeostasis is out the door. Just think for just the sodium potassium pump, which is a protein. If that one pump starts to denature, then we can't keep high sodium on the outside and high potassium on the inside. If just that one pump starts to denature, you will die from just that one pump. And there are lots of pumps and channels, receptors, anchoring proteins, enzymes. Any of those things start to denature, you can't maintain homeostasis on a cellular level. And if you can't maintain homeostasis on a cellular level, you can't do it on a tissue, an organ system, or an organism level at all. So all those things, pretty important. All right, so looking at homeostasis of extracellular fluid and intracellular fluid, right? If we think about fluid balance, the amount of water that we gain throughout the course of the day has to equal the amount of water that we lose over the course of the day. So minute by minute, it's not gonna be in homeostasis, second by second, minute by minute. But if you look over the course of a day, how much water you gain needs to equal the water that you lose. Then the question is, where do we gain most of our water? We gain most of our water through our GI tract. It's not the only place we gain water. Our cells make water when they do cell respiration. Water is a byproduct of cellular respiration. But most of the water that you gain is through the GI tract. And then where do you lose water? Well, there's a few ways you lose water, but the main place that you lose water is through the urinary tract. Yes, you lose it through sweating, you lose some through feces, but most of it's through the urinary tract. Now, keep in mind again, water can only move via osmosis. Water never uses pumps. Water never uses pumps. That means you can never actively transport water. Water only ever moves passively via osmosis. It can use water channels, but it's still diffusion through those water channels. Or it can just freely cross the membrane. Okay, simple diffusion. Electrolytes, same basic idea. If you look at your sum total of ions that you gain throughout the course of the day, sum total of ions, not individuals, but sum total, need to equal the number that you lose throughout the course of the day in order to be in homeostasis. Again, where we're going to gain most of those ions is through our GI tract, food and liquids. Where we're going to lose most of those ions is through the urinary tract. Now again, you can lose through sweat and feces, but most is through the urinary tract. And then acid base, same idea. The number of hydrogen ions that you gain throughout the course of the day have to equal the hydrogen ions you lose throughout the course of the day to be in homeostasis. Again, the big problem we deal with is making sure our pH doesn't get too low. The reason is because we produce more acids than we do bases. For example, lactic acid. If we are doing anaerobic metabolism, if we are making ATP without using oxygen, then we are doing glycolysis and the process of breaking down glucose, not using oxygen to make ATP, you produce lactic acid. We talked last time about if you have uncontrolled diabetes mellitus, or if you have not eaten in a while and your blood glucose levels are starting to get low, hormones will be released that not only try to raise your blood glucose, but hormones will be released that also start breaking down your stored fats. And when you start breaking down stored fats to get fatty acids to make ATP for glucose sparing, you will produce ketones. And ketones are acidic. Okay? So again, just cell metabolism, just kind of basic cellular activities, we tend to produce acids more than we produce bases. Now, where do we get rid of? Okay, so that's the primary place that we gain hydrogen ions, is through our own cellular metabolism. Things like this. Okay? Now, where do we lose hydrogen ions? There's two places we can lose hydrogen ions. Through the kidneys, and in particular, that's at the DCT. So we talked last time that we can reabsorb or secrete hydrogen ions at the DCT. And typically, since our pH is typically on the low side, typically we secrete hydrogen ions at the DCT. So we're getting rid of them. We're taking them out of the blood, putting them into the filtrate so that they end up in the urine, and out of the body they go. I'll say it one more time. So normally at the DCT, we're dealing with pH being too low, which means that we have too many free hydrogen ions floating around. And if that is what we're dealing with, which is what we usually deal with, then at the DCT, we will see an increase in secretion of hydrogen ions, meaning that we'll take hydrogen ions out of the blood that's in the peritubular capillaries, put it into the filtrate that's at the DCT. Those hydrogen ions go from the blood into the filtrate at the DCT. From the filtrate, they'll end up in the urine. Once they're in the urine, out of the body they go. Okay? We literally have eliminated hydrogen ions. The other place that we can eliminate hydrogen ions is through the lungs. So through the lungs, what we see at the pulmonary capillaries. At the pulmonary capillaries, we're going to have hydrogen ions that are in the blood, are going to combine with bicarbonate ions that are in the blood that's in the pulmonary capillaries. That then will cause carbonic acid to form. Carbonic acid is very unstable. Carbonic acid won't stay as carbonic acid for very long. It will either go back and form hydrogen and bicarbonate ions, or it will form CO2 and water. Now, what determines whether this reaction goes to the left or to the right is the concentration of CO2 and hydrogen and bicarbonate ions. So at the pulmonary capillaries, CO2 is diffusing out of the blood into the alveoli to be exhaled. That's what happens at the pulmonary capillaries. Oxygen's coming in, CO2's leaving. And so at the pulmonary capillaries, our CO2 levels drop. Okay? At our pulmonary capillaries, CO2 diffuses out and into the alveoli to be exhaled. And as that CO2 is diffusing out of the pulmonary capillaries and into the alveoli, that causes the partial pressure of CO2 to drop in the pulmonary capillaries. And as that partial pressure of CO2 is dropping, that drives this reaction that way, to the left. Okay, so this reaction is driven to the left as partial pressure of CO2 drops, and the CO2 is dropping because CO2 is diffusing into the alveoli from the blood to be exhaled. So since the CO2 is dropping, this reaction is driven to the left, meaning that hydrogen ions that are in the blood and bicarbonate ions that are in the blood are going to come together to form carbonic acid. That's going to break down into CO2 and water. That CO2 and water is going to diffuse out of the blood that's in the pulmonary capillaries, into the alveoli to be exhaled. So literally at the lungs, we are taking hydrogens that are in the blood and getting rid of them. They're just exiting the body as the hydrogens in your water. Okay? So this hydrogen here and that hydrogen here are these two hydrogens. And these two hydrogens end up being these two hydrogens that are being exhaled out of your body. So you eliminate hydrogen ions at the lungs, okay? Because that reaction goes that direction. So by changing your respiratory rate, you can get rid of more or less hydrogen ions. Okay? We'll come back and talk more about that. So the kidneys and the lungs are the two places we can get rid of hydrogen ions. Okay, so then this right here is just looking at gaining and losing water. You do not need to know any more about gaining and losing water than what we've already talked about, okay? I want you to know the major place you gain it is the GI tract, major place you lose it is urinary tract. You don't need to know numbers. You don't need to worry about the rest of that stuff. Okay, now hormones, okay? You guys already know these hormones. You've seen them now through the endocrine system and through the urinary system, and now you see them here again. I'm just gonna say a couple things about them that we haven't talked about yet, but it's still all with the same idea of what we already know about them. So these two hormones are the major hormones that help to regulate fluid balance in the body, ADH and aldosterone. They do it through different mechanisms, but they both are agonists. They work together to try to keep you from getting too dehydrated. These two hormones help you to reabsorb water so that your urine output is low, your blood volume stays high, and your blood pressure stays high. And again, when I say high, I don't mean it's high, I mean it keeps it in homeostatic range, right? It keeps it from getting too low. It keeps your blood from being too concentrated. So, ADH. ADH will be released when you have high osmotic concentration. When your blood has little water but lots of solutes, that's a high osmotic concentration. So that stimulus will cause more ADH to be released so that you retain water, you reabsorb water, and it helps to dilute out all that high osmolarity of your blood. It also causes you to feel thirstier because it stimulates your hypothalamus. We didn't mention that before. These two hormones are interesting hormones because they work in a variety of places. We've learned that they work at the collecting ducts, okay? We learned that last time. ADH causes more water channels to be inserted into the plasma membrane at the collecting ducts so that you reabsorb more water. The ADH also stimulates your hypothalamus so that you feel thirstier, and as you feel thirstier, you drink more water. If someone is drinking a lot, the hypothalamus gets inhibited, and the hypothalamus is where ADH is made. Not only can it stimulate the hypothalamus, the thirst centers, but it's also made at the hypothalamus. So people that are out drinking, ADH gets inhibited because the hypothalamus gets inhibited by alcohol. And when people are out drinking and their hypothalamus is inhibited and their ADH levels start to drop, they are reabsorbing less water. And since they're reabsorbing less, their urine output starts to rise. And people that are drinking, after about an hour or so, they run to the bathroom constantly, right? And that's because you're reabsorbing less water, you're urinating a lot, and it's also the one reason that you have a wonderful hangover the next day, because you're nice and dehydrated the next day. So it really is true. If you're gonna be drinking a lot, you should, for every drink you have, take a glass of water with it, okay? It helps to replenish all that water that you're not retaining because your ADH levels are dropping. Aldosterone. Aldosterone also helps you to reabsorb water, but it does it by influencing your sodium and potassium levels. ADH is going to cause more sodium to get reabsorbed, more potassium to get secreted at the DCT, and as you reabsorb more sodium, you will reabsorb more water. The water follows the sodium. So, aldosterone also, in addition to the things that we've already talked about, it also causes, and you can't see that last sentence here, but it also causes your taste receptors for salt on your tongue to become very sensitive, so that the taste of salt becomes very enticing. And as you start eating things that are salty, you start drinking a lot of water with it as well. So aldosterone also works in a variety of ways to get more water in your blood. Okay, that's just another way it does that. Okay. AMP and BMP. We talked about these guys too. Remember, AMP and BMP are antagonistic to ADH and aldosterone in terms of fluid levels. AMP and BMP are agonists, they work together, but these guys are only going to be released if you have severely elevated blood volume and severely elevated blood pressure. A little increase in blood volume and blood pressure, these guys aren't released, okay? So it has to be really high. What these guys will do is they will stimulate your central nervous system, your brain, to cause you to feel less thirsty. They also will inhibit the release of ADH and aldosterone, which are normally in your bloodstream. It'll cause less to be in your blood. And it will cause you to produce more filtrate at the kidneys. More filtrate means more urine volume. More urine volume means your blood volume drops. As your blood volume drops, your blood pressure drops. But again, these guys only released in severely high blood volume and high blood pressure. Okay, now, a few things about certain electrolytes. So, if you look across the membrane, let's get rid of this stuff here. Look across the plasma membrane, cell membrane, and you look outside of that cell versus inside of that cell, you're going to see certain ions at high concentrations outside and certain ions at high concentrations inside. And so there's a lot of charged particles in and outside of your cells. But there's certain ones that I definitely want you guys to know where they're high and where they're low, okay? Some of these we've already talked about, some of them we're gonna talk about, today or, Wednesday. So, let's do this. Okay, so if we look outside of cells versus inside. Okay, so outside would be our extracellular fluid, and inside would be our intracellular fluid. Okay, in and out, you've got lots of charged particles, okay? If we look outside, I want you to know that on the outside, you have high sodium. Now, does that mean you don't have sodium inside the cell? No, you definitely have sodium inside of your cells, absolutely. You just have a lot more out than you do in, but you have sodium in your cells. Outside of your cells, you have more chloride outside of your cells and less inside of your cells. That happens to be a negatively charged ion. Okay? Sodium's positive, chloride's negative. Also, outside of your cells, you have more bicarbonate ion outside of your cells and less inside of your cells. And again, that happens to be a negatively charged particle. Now, last time we talked, and we talked about pH at the DCT. What we said was, normally, at the DCT, we're dealing with our pH being too low, our blood pH being too low. And we said then that means normally at the DCT, we will see an increase in hydrogen secretion. We'll take hydrogens that are in the blood, where we have too many of them, we'll put them in the filtrate, so that those hydrogens will end up in the urine. So we'll end up with more hydrogens in the urine, and we'll have less hydrogens in the blood. Having less hydrogens in the blood means that our blood pH goes up, back to range, okay, back to range, back to homeostatic range. And having more hydrogens in our urine means that our urine pH goes down. Okay? So if we look at our blood pH should stay about 7.35 to 7.45. Okay? That's where you want it to be. So if our blood pH becomes too low, that means that it is less than 7.35, and we want to get it back into range, 7.35 to 7.45. And as we're doing that, then those hydrogens end up in the urine, and so a typical urine pH is around a 6 or so, something like that. Okay? It can go all the way down to 4.2 or something like that. But usually sits around a pH of 6-ish. Okay? Now, what I also said was that you used a certain kind of transport mechanism, certain kind of pump. And whatever you did to hydrogen at the DCT because of pH, you did the opposite to bicarb. And I said that when we started talking about this stuff, that you would see the significance of bicarbonate ions and why we want to be doing the opposite normally, which is reabsorbing. So if we are secreting hydrogens at the DCT because our pH is too low, that means we're also doing the opposite to bicarb. So that means we would be increasing bicarbonate reabsorption. So that's normally what we do, okay? And as we're increasing bicarb reabsorption, that means that we will have more bicarb going in the blood, and less bicarb will end up in the urine. have more bicarb in the blood, what we're really talking about is the plasma, the liquid part of the blood. And I had mentioned before that the plasma is part of the extracellular fluid. And so now I want you to notice that typically we have a high concentration of bicarb in our extracellular fluids. Part of the way that we have all that extra bicarb is because this is what we tend to do with the DCT. Now there's other ways to get bicarb in our extracellular fluids. We'll talk about that later. But this is one important way that we tend to keep a lot of bicarb extracellularly. Is at the kidneys, we don't let it go. We don't put it in the urine, we put it back into the blood, and blood is part of our extracellular fluids. Think about when you look at a regular old systemic capillary, right? So this is your capillary, and in here is your plasma, and all this out here is your interstitial fluid. Remember, we had all that water and solutes come out of the plasma into the interstitial. That's all extracellular. Your extracellular environments communicate with one another. You've got water and fluids coming out, you've got it coming right back in. So that bicarb that was in your plasma, it will end up being out here in your interstitial fluids, all things right there, extracellular. Okay, we'll come back and talk about why we want so much of this outside later, okay? We'll see why later. But that's what we got, okay? Now, if we look inside of our cells, there are certain things that we have at high concentrations inside of our cells relative to the outside. Potassium is a biggie. We've got more potassium in. We certainly have potassium outside of our cells, but we have a lot more in than we do out. We have more magnesium inside of our cells, less outside. And we have a lot of negatively charged proteins. So these are dissolved proteins. And at a kind of resting pH, these proteins, these dissolved proteins, tend to have negative charges on them. So inside of our cells, we have a lot of dissolved proteins. Just think about all those enzymes we were just talking about. Enzymes catalyze reactions. Reactions happen inside of cells. Enzymes are water soluble. So here's some of those dissolved proteins are enzymes. Okay? Plus there's a lot more. So you've got a lot of proteins inside the cell that are dissolved. You have very little outside. Okay? There's no point for them to be out here. They can't do much out here. The other thing that we have a lot inside of our cells is we have a high concentration of monohydrogen, so one hydrogen, monohydrogen phosphate. PO4, is that 3 minus? No, 1 minus. Monohydrogen phosphate, okay? We're gonna come back and talk about why that's a big deal later. Now, these are the ions that I want you to know where they're high and where they're low. These are not the only ions that are high on one side and lower on the other side, but these are the ones I want you guys to know. Now again, I also want you to keep in mind that even though individual ions can be high in one place and low on the other, if you add up all of the solutes outside and all of the solutes inside, they need to be equal. Right? So the sum total solutes outside of your cells needs to be equal to the sum total of solutes inside of your cells. Okay? So individual ones can be high somewhere and low somewhere else. But when you add all of that stuff up, that stuff plus glucose and amino acids, all of the solutes, they need to be equal, because if they're not, you will have fluid shifts. Water will start moving towards where you have a greater concentration of total solutes. You don't want that. Okay? You don't want that. That's what this is saying right here. Osmotic concentrations. So osmotic concentrations is solute concentrations. Osmolarity or osmotic concentration is solute concentration. So that's saying that your osmotic concentration, the concentration of your solutes in versus out, need to be equal. If they're not, so that's saying they need to be equal even though individual ions can be high versus low. Sum total has to be equal, otherwise osmosis will occur. And water will move either out of the cell if the sum total's higher outside, or osmosis will cause water to come in if the sum total inside is greater. And we do not want that. Okay? I will definitely ask you something about that in the test. Okay, so then making sure that we remember these terms. Hypotonic, hypertonic, isotonic, okay? We're gonna review that, and that's looking at this right here, okay? Where does water go if we're not in an environment where in and out have some total equal osmotic concentrations? What's gonna happen? All right, so let's look at this. So I'm going to put 3 extracellular environments up here. Here, it could be interstitial fluid, it could be plasma, it doesn't matter, it's just extracellular. Here's another extracellular environment right there, and I'm gonna do another one right here. Okay. And in each of those extracellular environments, I'm gonna stick a cell. I'm gonna put a red blood cell in here, but it doesn't really matter. So if I put a red blood cell in here, then that tells you this is the plasma of the blood. Again, it doesn't really matter. What really matters is that we have extracellular and intracellular environments. Okay, now we're gonna look at the tonicity of these environments. Okay? We're gonna look at the tonicity of the extracellular fluids. So this extracellular fluid, I'm gonna make this one hypertonic. Hypertonic extracellular fluid. I put that there just to remind you that I'm not talking about inside the cell. Because I could say hypertonic intracellular, and that would mean something very different, okay? So typically when I use these words, I'm talking about extracellular, but I'll try to always clarify so you don't have to assume anything, okay? Over here, we're gonna make this a hypotonic extracellular fluid. And then over here, we're gonna make this an isotonic extracellular fluid. Now, let's define each of those, and then we'll put some real numbers here and see what happens with water. Hypertonic. So what this means is the solute concentration, so the osmolarity, okay, the solute concentration or the osmolarity outside of our cells, so that means extracellular fluid outside, is hyper or greater than the solute concentration or osmolarity inside of our cells in the intracellular fluid. Okay, hypotonic is just the opposite. Here, hypotonic, we've got the solute concentration of the outside of our cells, the extracellular fluid, is hypo or less than the solute concentration or the osmolarity of the intracellular environment or inside of the cell. Iso means equal. So isotonic means that our solute concentration outside or extracellular is equal to the solute concentration inside of our cells or in the intracellular fluid. So let's do some real numbers here. Okay, so what I'm gonna do just to make things nice and simple is, let's just talk about sodium chloride, okay? We won't worry about all the other solutes, let's just look at sodium chloride. So if you look inside of your cells, and you look at sodium chloride, you have about a 0.9% sodium chloride solution inside your cells. I'm just gonna put that for all these cells, 0.9% sodium chloride, 0.9% sodium chloride. Now, instead of saying percentage of something, I could also say something like a 2 millimolar, or no, I could say 2 milliosmol sodium chloride, okay, or something like that. It's just a concentration of a solute, okay? I chose to do percentage, but I could also express it in milliosmoles as well, okay? Same basic idea. I like doing percentages because I can think about it like this. I can say, if I'm making this real simple and I'm not worrying about any other solutes like glucose and amino acids and potassium and all that other stuff, then I could say, okay, if I've got this, that means I've got 0.9 parts of sodium chloride to 99.1 parts water. When you add those two up, it equals 100. 100%. So over here, I've got 0.9 parts stuff, sodium chloride, to 99.1 parts water. And the same thing here, I've got 0.9 parts stuff, or sodium chloride, solute, to 99.1 parts water. So then, before I start doing these kinds of problems, I always just write up in the corner what osmosis tells me. Osmosis says that water will move from areas where I have high water concentration, low solute concentration, to areas where I have low water concentration, high solute concentration. Okay, that's what osmosis says is going to happen. So, okay. And then I just start putting real numbers in to see what happens. Okay, so if I come over here and I say, okay, I want an extracellular fluid that is hypertonic to my cell. So this means that my solute concentration out here has to be greater than the solute concentration in here. And here it's 0.9% sodium chloride. So that means out here, I've got to have a sodium chloride solution that's greater than 0.9%. So I could make it 0.91% would be hypertonic to that cell. That would be a small difference between the two environments, and I wouldn't get a lot of water moving. The greater the difference, the more the water will move. So I'm just going to make it really exaggerated. Okay, I'm going to make it real big, and I'm going to say out here, let's put a 9% sodium chloride. That would not happen in your body. You'd die before that happened. Okay, but let's just say that's what we're dealing with. So this is 9 parts sodium chloride to 91 parts water. Right? So again, this is a hypertonic extracellular fluid because my solute concentration out here is 9%, and in here it's 0.9%. So it's greater out, lower inside. And then I say, where's water gonna go? Okay, so then I look at this and I say, okay, I'm gonna assume that that cell's not permeable to sodium chloride, so I don't have to worry about sodium chloride diffusing across the membrane. And I know that water is permeable to any membrane, so I know water can freely move. And I'm gonna tell you that that cell's not permeable to sodium chloride, so we don't have to worry about it moving. So then I say, okay, water's gonna move from low solutes to high solutes. Low solutes, 0.9, high solutes, 9. Water's gonna leave that cell. Or I could say, osmosis says that it's gonna go from high water, low solutes, high water, 99.1, to low water, 99, or low solutes to high solutes, 0.9 sodium chloride to 9 sodium chloride. Okay? So water is going to leave that cell. If you put your cells in hypertonic environments, water will leave that cell. So water will diffuse out. And the greater that concentration difference, the faster and the more water that will leave. In that environment, our cell will shrivel up really fast. If it were 0.9 out here, 0.91, you'd have such little water moving that you really wouldn't even notice it. But something like that, so much water is going to leave that the cell will shrink. And when you look at a red blood cell shrinking, that's called crenation. A red blood cell shrinking is called crenation. It's not critical, you know that word. What's critical is that you can figure this out. Over here, we do the opposite, hypotonic. This just means that outside will have less solutes than inside. So that means outside, I just need to have anything less than a 0.9% sodium chloride. So I could make it 0.89 would be hypotonic to my cell. I can make it a 0.1 sodium chloride. That would be hypotonic to my cell. Again, I'm going to exaggerate it and make it as low as I possibly can and make it a 0% sodium chloride solution. That definitely is hypotonic to that cell. Anything less than 0.9 would be less solutes out compared to in. And if I have 0% sodium chloride out here, that means I have 0 parts sodium chloride, which means I have 100 parts water. And that's what distilled water is, right there. That's distilled water. Again, it would be very hard to get your body at that point. It's doable, but you wouldn't want it to happen. You would not feel good if that happened. So here we say, okay, if that cell is impermeable to sodium and chloride, but it's permeable to water, because water can go wherever it wants, basically, then we say, where's water gonna move? Water's gonna move from high water, low solutes, to low water, high solutes. So here we've got high water, 99.1, to low, high water, 100, to low water, 99.1. Or we could say water will go towards high solutes. Water will go from low solutes 0 to high solutes 0.9. If your cells are in hypotonic environments, your cells will gain water. So here your cells will gain water. This could cause your cell to swell. And if enough water entered, because the concentration difference was great enough, you could get that cell bursting. A red blood cell that bursts, that's called hemolysis. Hemolysis is when red blood cells burst. Obviously, you don't want that happening, because then they can't transport oxygen anymore. So you don't want either of those situations. You don't want your cells gaining water, and you don't want your cells losing water, because that one can cause your cell shape to change dramatically. Sometimes the shape of the cell is real important. When we talked about red blood cells, it was real important that they had that biconcave disc. That was real important. And here, if it starts changing that shape, it's not going to be able to transport oxygen as efficiently. You also don't want your cells gaining or losing too much water, because it's going to alter all the other solute concentrations in there, and that can mess up your cell metabolism. That can mess up the concentration of reactants and products for chemical reactions. And again, your cells are just little chemical factories. So you don't want those things happening. What you want your cells in isotonic environments. So this would be where you have equal in versus out. And if you have equal in versus out, your net movement of water is 0. So if you go to the hospital, and depending on what's wrong with you, but let's say that we're not dealing with too many electrolyte issues, okay? You go to the hospital and they're gonna put an IV in you and they're gonna hang a bag of normal saline. That's what this is. Normal saline is a 0.9% solution because it is isotonic to your cells. If you see someone hanging a 9% sodium chloride solution or a 0.09% sodium chloride solution, you should ask them, why are you doing that? Because if it were a 9%, then that would be this environment here. And if it were a 0.09%, that would be this environment here. Now, there may be a justifiable reason for that, but I would want to know that before I saw that hanging up in my IV, right? This is what you want your cells in. The other thing to kind of think about here is when you're dehydrated versus overhydrated, okay? So when you're dehydrated, you have too little water in your extracellular fluids. So that would be this environment here. Okay? This is what happens when you're dehydrated. When you're dehydrated, you're not taking in enough water, and if you're not taking in enough water, out here, your water concentration will drop, and as your water concentration drops, your solutes become more concentrated. So here you have more solutes relative to water, and that's a hypertonic environment. Okay? If you're dehydrated, your cells will try to compensate by having water come out to try to fix this outside environment. And it would be better if you just drank some water, okay, as opposed to that happening. If you are overhydrated, then this can occur. Now, you have to really drink a lot of water to overhydrate, but it can happen, okay, but that's what's happening here. You have too much water outside of your cells, causing the solutes to become too dilute. Your solute concentration drops because your water concentration is going up. Again, you want this. Okay. There will definitely be 1 or 2 of these multiple choice questions on your test. Okay, so then this is looking at what we just talked about. This is talking about dehydration. Again, what I want you to know is what we talked about. Okay, anything else that's on here you do not need to worry about. Okay. Now we're going to start talking about pH. Okay. I'm going to take a sip of water. I'm going to erase that board, and then we'll start in on pH and buffers. This clock is right. Okay, so we will go to about 8:15 is kind of where I'm shooting for. 8:50, we'll leave it at 8:50?
I know, I thought you guys were gonna have a break until 8:15, I was like, whoa.
Alright. If you guys need a break before this, you let me know. Otherwise, we'll just plow through this. Alright, we're gonna erase this stuff. We're gonna do some review about what is pH. We're gonna talk about different kinds of acids, and then we're gonna set up what is a buffer. Okay? So that on Wednesday, what we can do is we can talk about buffer systems. We'll talk about our acid-base disorders. It's kind of where I'd like to get us. And I'm trying to keep any one single lecture from being too horrible like I did last time. Alright, so. Let me see if I can get a better marker. Give me 1 minute here. Homeostatic range for pHs. We now are going to learn the homeostatic range for CO2 levels. So, the homeostatic range for CO2 is to have a partial pressure of CO2 between 40 and 45. If the partial pressure of CO2 goes below 40, partial pressure of CO2 goes below 40. So if I tell you your CO2 is less than 40, what's that going to do to your pH? So if your CO2 is becoming too low, that means that you have more or less hydrogens floating around in your blood. Less. And if you have less hydrogens floating around in your blood, what's that do to your blood pH?
Goes up.
Causes it to go up. So now we start putting numbers with it. Okay, we already put our pH numbers with things here and here. And now we're putting our CO2 numbers and looking at the pH. So if the partial pressure of CO2 is less than 40, then we are going to end up being in an alkalotic state. Our blood pH is going to end up being greater than 7. And you can say the opposite too. If the partial pressure of CO2 becomes greater than 45, normal homeostatic range, millimeters of mercury. If that's out of homeostatic range, then that means that now we have too many free hydrogen ions floating around. And if we have too many free hydrogens floating around, that means our blood pH is going to be dropping. And if this is out of homeostatic range, then our pH is going to be out of homeostatic range. So that means we're going to be less than 7.5. And in an acidic state. So, even though CO2 is a waste product, it can't get too low. If your CO2 levels get too low, it will cause your blood pH to get too high, and you can't have that. Okay? So, even though CO2 is a waste product, you can't let it get too low. If CO2 didn't directly affect pH, then maybe you could get rid of CO2. But you can't get rid of all of it. And CO2, like pH, stays in a pretty tight range. Okay, we'll see this equation again. Okay, so then let me just see. Okay, I'm going to do a couple more slides and then we'll stop. Okay, so I had said before that to homeostatically regulate pH, okay, we're going to be talking about pH for the last 15 minutes today and all of most of Wednesday. Okay, that's what the rest of this PowerPoint's about, pH and acid-base disorders and buffers. So I had said to homeostatically regulate pH, you needed to make sure that the hydrogens that you gained throughout the course of the day equal the hydrogens that you lost throughout the course of the day. And I said that the way that you gained hydrogens was through just cellular metabolism, and the way that you lost hydrogens was either through the DCT of the kidneys or the lungs by exhaling CO2. Now, we then need to think, okay, that's great. That's great, that's how we're gonna homeostatically regulate it. But what are we gonna do while those hydrogen ions are traveling through the blood, going to the kidneys and to the lungs? We can't just let those hydrogens freely float around in the bloodstream until we can get rid of them at the kidneys or at the lungs, because our pH would be way too low if we just let them freely float around till we got rid of them. So we can't do that, okay? We have to do something on a short-term level, a temporary fix, until we can get rid of these hydrogen ions at the lungs, at the kidneys. And that temporary fix, buffers, okay? Buffers are a temporary fix to a pH issue. They are not a homeostatic fix. And those words are gonna be real important on your test, okay? So if I say temporary fix, I'm talking about buffers. If I'm talking about homeostatically correcting a pH issue, now we're looking at what's going on at the DCT of the kidneys and at the lungs, okay? All right, now, how do buffers work? What a buffer is a compound, okay? A buffer is a compound that can do one of two different things. It can either suck up hydrogen ions from the solution, or that buffer can release hydrogen ions to the solution. Depending on what the pH is, that buffer can either suck up hydrogen ions, so they're not floating around free, or, depending on what the pH is, that buffer can release hydrogen ions, so there are more hydrogen ions floating around free. Now, let me erase this, and I'll say a few more things about that. So, before we get into more details with the buffers, I want to just define an acid versus a base, and then talk about what makes an acid strong versus weak, okay? And then we'll clarify what that buffer is, and that's where we'll be stopping for today. Okay, so first of all, what an acid is. An acid is anything that can donate hydrogen ions to the solution. So for example, when we talked about carbonic acid, okay? Carbonic acid was H2CO3, that's carbonic acid. And what carbonic acid can do is it can disassociate and donate hydrogen ions to the solution that it's in. And then we also get bicarbonate ions being released as well. But the fact that this happens right here, the fact that those hydrogens are released or donated to the solution, that's what makes that an acid. Okay. Let's take hydrochloric acid. Hydrochloric acid is HCl. If I put hydrochloric acid in water, what it's gonna do is this. It's gonna break apart into hydrogens and chlorides. It just donated hydrogen ions to this solution that it's in. It's an acid. That's the definition of an acid. If it releases or donates, okay? So you could also say release. If it releases or donates hydrogen ions to the solution that it's in, then it's an acid. Okay? A base absorbs hydrogen ions. Okay? So a base will absorb hydrogen ions from the solution. So if I wrote this, if I wrote hydrogens plus hydroxide, okay, and then I did this. This hydroxide is acting as a base because it just absorbed that hydrogen ion. It took it out of the solution and bound it up. So right there, hydroxide would be a base in that situation. Now, let's think about the difference between a strong acid versus a weak acid. A strong acid. I'm gonna take HCl, okay? HCl is a strong acid. What that means is when I put HCl in solution, let's say I put 100 molecules of HCl. So I've got 1 HCl, 2 HCls, 3 HCls, let's say I have 100 of those, and I drop them into water. Every single one of those will break apart. That's a strong acid. Strong acids will release a lot of hydrogens to the solution. So if I dump a strong acid into a beaker of water, all of it will break apart. So I'll end up with 100 hydrogens floating around and 100 chlorides floating around. If HCl were a weak acid, it's not, but let's just say it was, okay? If HCl were a weak acid, then not all of them would break apart like this. Only some of them would. So if HCl were a weak acid and I started with 100, I'm making up numbers here, okay, so I don't know exactly what the number would be, but only some of it would release hydrogens. So maybe only 10 would be released, which means that you would have 10 chlorides, and the other 90 would not have broken apart. That would be a weak acid, where it only releases some of its hydrogens, and it keeps the rest. Now, I differentiate these two because buffers act as weak acids or weak bases, meaning that if a buffer is acting as a weak acid, it will release some of its hydrogens, but not all of them. If a buffer is acting as a weak base, it will absorb some hydrogens, but not all of them. So a buffer acts as both a weak acid and a weak base, depending on what the pH is. It can release some of its hydrogens, but not all. It can absorb some hydrogens, but not all. And that's what makes it a weak acid versus a weak base. If it's releasing some hydrogens, it acts as a weak acid. If it absorbs some hydrogens, it acts as a weak base. Now, I'm gonna put an equation up. If you understand this equation, then the 3 buffer systems that we're gonna start talking about on Wednesday, you can put all these buffer systems into this basic equation. If you understand the basic equation, you should be fine with all these buffer systems. So let's do the basic equation and we'll do the buffer systems on Wednesday. So all buffer systems will work like this. And again, we're gonna talk about 3 of them. So what we're gonna do is we are gonna take hydrogen and put it with something, and we're not gonna worry about that, what that other part is. We're just gonna put a Y there as a symbol, meaning it could be a variety of different things. So the H is hydrogen, and the Y is just a symbol for something else, okay? Now, this right here, this HY, when it's together as HY, so H and Y, when they're together in a solution, it can act, it is a buffer, and it can act as a weak acid, meaning that this thing, this buffer, can release some hydrogens, not all of them, but some hydrogens can be released. And then what also will inevitably get released are the corresponding anion. The anion is the negatively charged part. So here, this buffer, when we write it like this is acting as a weak acid because it's releasing some hydrogens. And And then when you release some hydrogens, you also are releasing some of the anion. But then the same buffer can act as a weak base, and what will act as the weak base is this part right here. So that can act as a weak base. The anion can act as a weak base, which means that it can absorb hydrogens. So here it will absorb some hydrogens. And when that anion, acting as a weak base, absorbs some of those hydrogens, then we form the HY. Okay? So all buffer systems are going to work like this. If we are at a neutral pH, then this reaction is completely balanced. Meaning that if you look in your solution, you will have equal amounts of HY to equal amounts of H and equal amounts of Y. It'll all be balanced. I think of it like a straight teeter-totter. But if the pH starts to shift, if the pH of that solution gets too high or too low, then that chemical equation that's flat right here is going to start shifting one way or the other. It's not going to completely shift one way or the other, but it'll shift a little bit one way or the other. So, for example, if the pH of this solution starts to drop, okay? First thing I always say when I see something like this is I put in parentheses, that means I have too many free hydrogens in that solution. So if that's what's happening, to temporarily fix the pH issue, now we gotta think about what to do. If the problem is that the pH of the solution is a little bit too low, we have too many hydrogens, then do we want our buffer, okay, we want our buffer, and we're going to talk about different buffer systems on Wednesday. We want our buffer to act more like a weak acid or a weak base. If it acts like a weak acid, it will release some hydrogens to the solution. If it acts like a weak base, it will absorb some hydrogens from the solution. What do we want it to do if this is the problem? We want it to act as a weak base. We want it to act more like a weak base. So, if we do this, so we've got our equation here, okay, HY, H plus Y, and we said our pH has dropped, which means we have too many hydrogens floating around. So this is what's going on. We've got too many of these that caused our pH to drop. So then to fix the problem, we need our buffer to act more like a weak base, which means we need this reaction to start shifting this direction a little bit more. We need our weak base to start absorbing some of those hydrogens so that we start producing some of that. And as we start producing more of that, we will get rid of some of those hydrogens because they will be bound up now. So, on your test, I might say something like this, okay? I would map it out like this. I'd say, if your pH of your solution dropped, to temporarily fix the problem, you would want your buffer to act more like, I might ask you that, and you would say more like a weak base. Or I might say this, to temporarily fix the issue, you would see, and then I would say more or less free hydrogens. more or less free anions, more or less of the weak acid. And the way I work through this, I say to temporarily fix the problem, I would want this to act more as a weak base. The reaction would go more to the left, not completely, but more. It would start to be like a teeter-totter, start to tip a little bit to the left. And as this reaction tipped more to the left, and as this weak base here started absorbing these hydrogens, what I would end up seeing is less free hydrogens floating around in that solution, less of the weak base floating around in that solution, and I would end up seeing more of the weak acid. As my weak base absorbed those hydrogens, this is what I would end up seeing. If that makes sense to you, you will be fine with these questions. If that doesn't make sense to you, you just need to walk through it and think about it. So, opposite pH, opposite happens. So I'll write this out, but if you understand this, then it's just the opposite, right? So up here, we're gonna say if the pH started to become too high, or I could say something like, well, we'll leave it at that right now. We'll get into more details later. If the pH of the solution became too high, to temporarily fix the problem, you would want your buffer to act more like, and again, I always look at this and I say pH is too high, it means I have too few free hydrogens floating around in that solution. So to get more free hydrogens floating around in that solution, I would want my buffer to act more like a weak acid and release hydrogens. So that means I would see this reaction, this would be my weak acid. I would see this reaction go more to the right, releasing more of these, so those would increase. As you get more of these, you're gonna end up with more of your weak base, or your anion. And as you have more of each of these, you have less of that by the time you're done. Okay? And that's literally how I go through every single one of these. I do it like that. We're gonna go through 3 separate buffer systems and we're gonna learn their equations, but you always can break it right down into HY, HY. Every single one of them, just write it down and work out. Okay, that's where we're gonna stop for today. We'll finish this on Wednesday. Again, if that doesn't make sense, try to review that before